Thank you, Dean Malavala. Congratulations, graduates. You have reached a milestone at a particularly difficult time. You've persevered through a pandemic, and it is a very unsettled world. I am deeply honored to be here with you, your families and friends, as well as faculty and administrators to celebrate your accomplishments. I'd like to share some of my family history and a couple of stories about the time after I left MIT, a place I considered at the time, and still do, as graduate school heaven. I felt that way because the faculty, the staff, other students, and postdocs welcomed us as junior partners in important endeavors. Of course, our more experienced colleagues were blunt in letting us know when we were ignorant, missed the mark, or performed below expectations. Then, as now, the bar was high. Each of you has a history that ultimately led you to MIT and to this ceremony today. Cherish that history. Honor the people whose actions resulted in the unique person that you each are. And even if you don't know the details of your story, you're here. And that serves as proof that there is a story and you continue it. My 5 siblings and I all resemble my father, the son of an indigenous Mexican Indian tribe unknown. My mother was a blue-eyed, red-headed, fair-skinned descendant of the Spanish conquistadores from the Basque region of Spain. Over half of my genetic makeup can be traced to Spain, while under 40% is attributable to the indigenous peoples of Mexico. The rest is a sprinkling of Arabic, African, Asian, Jewish, and a sprinkling of Neanderthal throughout. My paternal grandparents, Encarnacion and Luz Villa, came to this country during the Mexican Revolution. My father grew up in a house with no running water and a dirt floor that my grandfather and his older sons built. My father was one of 15, a middle child. Only 10 survived to adulthood, and all worked in the fields as children. My mother's grandfather, Manuel Larrañaga, and his wife, Cleofas, owned a ranch in Arizona. He sent his daughters to get a teaching degree because he didn't want them to marry cowboys. His sons had to stay on the ranch and be cowboys. Both of my parents grew up speaking Spanish, but because my parents had been punished for speaking Spanish in the school playground, they decided that their children would grow up speaking only English. As a result, I felt I was somewhat an outsider in my very large extended family, which turned out to be good preparation to be part of an enterprise where I did not fit the standard model. Towards the end of graduate school, I arranged a postdoc at the institution up the road and confidently began a project to isolate genes coding for proteins that make up the eggshell of the wild silk moth. Now, this was well before many of the tools of molecular biology were available. There were no kits. Restriction enzymes, as well as many other important reagents, were not commercially available. Phages weren't used as cloning vehicles, and DNA sequencing was in development. And there were no cell phones. Much of the public and some scientists were worried that mixing DNA from different species would result in an uncontrollable plague. So we decided that I would go to Cold Spring Harbor, where recombinant DNA technology could be conducted. Now, I had chosen an important problem. How are genes regulated during development? But it was not clear that it was doable with the model and the tools that I had at hand. I was working alone. I didn't ask anyone for much input. Would it have changed anything? I can't say for sure. But what I know is that I spent a year in Cold Spring Harbor plating out bacteria that I hoped had eggshell genes and waking up day after day to nothing. So I was trying to do not the impossible, but I was trying to do not the improbable, but the impossible. The polyphemus genome, the silk moth genome, was too big. The fragments were too big. The efficiency of each step was low, and the bacteria that I had to work with was very badly debilitated that had to be used under stringent conditions. There was no way we could get any clones whatsoever. However, I left Cold Spring Harbor with an aliquot of every enzyme in Rich Roberts' freezer, a very valuable gift. That freezer was the origin of New England Biolabs, still a major supplier of restriction enzymes and other reagents. So I returned to Cambridge. No project, no papers, a valuable set of enzymes, shaken confidence. On my return, Arjun Stratiatis, my colleague in the lab, invited me to join a project he was doing with Wally Gilbert. The objectives of that project were to clone the rat proinsulin gene to achieve expression of the gene in bacteria, and then to do the same with human proinsulin. Rat insulin was first because we couldn't work with human DNA in the United States at that time. The project was obviously important, both scientifically and commercially. The team had a rich source of rat insulin RNA from a pancreatic tumor we got from colleagues at the Joslin Clinic. Members of the team had all the skills, almost all the skills needed, but they still needed someone to put the DNA into a plasmid and put that into the bacteria. Well, while my time at Cold Spring Harbor had been completely unsuccessful in terms of successful experiments and papers, I had learned how to manipulate DNA, and I had learned how to handle that finicky bacterial strain. And I had a treasure trove of enzymes not generally available, and I really didn't want to end my postdoctoral career with an unsuccessful project. Lessons are learned from failure. Don't forget that. Now, I didn't meet Wally until we'd started to get results. Arj talked to Wally all the time. I talked to Arj all the time. The goal was clear. We knew what had to be done, and Wally left us to it. This time, we talked and argued about our approach. When necessary, we called colleagues with special expertise for advice. We thought through every possible difficulty, and we were lucky. Lucky is the wild card in life and science. Cherish it, but don't count on it. Within 6 months, we had an insulin clone that resulted in the production of a minuscule but momentous amount of insulin. When it came to the last goal, human insulin, we had to go to a high containment facility in England. We had to get dressed in hazmat suits. We had to shower in and out of the laboratory. All manipulations had to be done in a glove box, which was a part of the room with thick gloves going into that space through a transparent window. Again, not much worked. So 4 weeks later, we were very frustrated, and we came back empty-handed and knowing that the team at Genentech had beat us to the cloning of human insulin and also to its expression. Nevertheless, we were part of the birth of biotechnology. Biogen had been founded several months before we went to England. I declined a position there because both my graduate work and my postdoc work had been done at some of the best labs in the world, and I wanted to see what I could do on my own. I did, however, become a consultant to Biogen and had a front row seat as Biogen grew over the next 8 years. It was a great tutorial and business and came in handy later when I became chair of the board of TKT. That's a story for another time. I returned from England to a position at UMass Medical School in Worcester as an assistant professor and set out to build a career. In an academic position in a research institution, the key measure of success is research, and research productivity is measured by grants and papers published. I knew this. Nevertheless, I took on too many service jobs, extra teaching, more students, many committees. I didn't focus on my research plan, and so, as a result, the tenure process, which in academia is always never fun, was particularly painful for me, and it was a squeaker. I got tenure and left UMass 1 year later to accept a non-tenured position at Harvard Medical School and Children's Hospital. My reasoning at the time was that I would go to a more rigorous institution. Clearly, I hadn't yet realized that one's research depends on one's own efforts with your colleagues, as long as the institution has reasonable resources available to you. My move was the right one for me, but I made it for the wrong reasons. So you can do the right thing for the wrong reasons. I was at Children's Hospital and Harvard Medical School for over almost a decade. I had great colleagues, students, and postdocs, and we published on a wide variety of subjects. Many of those papers continue to be published in the current literature. We used the tools of molecular biology to investigate the development of vision, the effect of amyloid peptide on neuronal cells, secretion of insulin, the DNA sequence of several genes, development in the human brain, and the role of a couple of insulin-like growth factors. Each question was interesting and important. However, focusing on so many different topics and not following up on initial findings was not the best way to gain professional recognition. After 10 years, I decided it was time to explore my options. Initially, I thought I'd go back to the lab full-time because I really loved it. But after consulting with many people, it became apparent that I had strong administrative skills, so I began to explore those options. I moved to Northwestern University as Associate VPR, and 2 years later, after a nationwide search, I was appointed Vice President. It was a wonderful time to be at Northwestern. There were sufficient resources that I could say yes more often than I said no, an ideal position to be in. And we made great progress during that time under the Bienen administration. Now, while at Northwestern, I had tried to recruit Susan Lindquist from the University of Chicago to Northwestern. At the same time, Sue was being courted to come to the Whitehead Institute here in Cambridge. After she got here, she recruited me to become the COO and Vice President for Research at the Whitehead. And while I was there, the main job was to move the Genome Research Center, which was a Whitehead activity, to an independent center affiliated with MIT, Harvard, and the Whitehead. After 3 years, Susan decided that her heart and mind were focused on her scientific work, and she returned to her laboratory as a Howard Hughes investigator. I left Whitehead and joined Cytonome. That company had been started by John Gilbert to develop the instruments you heard about to sort human cells very rapidly under sterile conditions. My intent was to stay for a year, but I stayed 8. John left to start other companies, and I became CEO and had to find money in 2008, just as the economy was coming to a crash. During that time, the executive team took no salary. I had to lay off half the staff, and I made over 200 pitches to no avail. It was much harder than my time at Cold Spring Harbor. Then, in 2009, an opportunity arose from a most unexpected source. A company based in Texas, now called Genetics ST, was using conventional cell sorters to produce bovine sperm enriched for the X chromosome. This sperm was sold to dairy farmers because dairy farmers have to replace their milk cows about once every 3 years. They don't have so much use for the male calves. So this company was on the rise. The CEO of that company, Juan Moreno, liked our technology, and so he decided to invest, and John Sharp, who had vast experience building cell sorters, took over as CEO. I stayed on as CSO and on the board until 2014, when I left the company but not the board. At present, Cytonome still supplies Genetics ST with sorters, and they are now spread all over the world, working 24/7. And the Gigasort is in use in Japan for a clinical trial conducted by Sumitomo Pharmaceuticals to sort differentiated neuronal cells to treat Parkinson's disease. We think that a trial will start in this country soon. After I left Cytonome, it took me a little while to decide how I wanted to spend my time. Ultimately, I decided to start a one-person consulting company to serve as an umbrella for my activities. I had always been puzzled by how slowly women and people from historically marginalized groups were incorporated into science, technology, engineering, math, and medicine. Many people I knew in these fields seemed to agree that more diversity was needed and desirable. Well, a friend of mine, Florence Bonner, who is VPR at Howard University and a social scientist, said to me, you know, you natural scientists tend to talk about diversifying STEM, but you aren't considering any of the things that we, the social scientists, have learned That made sense. So I began to look into studies on human decision-making, including the role and neurological underpinnings of unconscious bias. Bottom line, unconscious biases are universal. All of us have them. It's a big data problem. Our minds, as wonderful and as complex as they are, has to have shortcuts because it cannot deal with the amount of data we are receiving all the time from all our senses. I developed a formal presentation when the program on science, technology, and society at MIT awarded me the Elting E. Morrison Prize in 2016. My role now is to provide a dispassionate, data-driven explanation for why well-meaning people have not been able to improve the diversity of STEM fields. My goal is to open minds, especially at the decision-making level. One lecture will not change any minds, but it might make it easier when someone else at the institution suggests some processes to ensure that implicit biases mean that individuals are not eliminated before their credentials are evaluated. As of this writing, I've given about 80 or so presentations to universities, colleges, companies, and federal agencies. My degree is in cell and molecular biology, and it's enabled a career, though, in many different fields in a whole bunch of roles. That's what you've gotten with your degree, the ability to learn, to think critically, to be persistent. The specifics of your study will become less important as you proceed through your career. In other words, your degrees are not degrees that define limits. It is a key to doors, and which doors you go through are up to you. Thank you, and congratulations once more.